This is just answer 10 questions. We don't even have to
And
write our own storyline.
yeah, God bless. I was like, I'm so glad they finally tell us like just answer the question on the card. If only you could have done that for the other one too.
Yeah, definitely. But the one thing that I'm bothered by kind of now is question 10. Just write down in this big ass table with half the shit that's there. You don't even have to do, but write down who's done what.
Yeah, I hate those things, man. Like,
Dude, most of the time we're just next to each other, running in one computer.
yeah, like, and not only that, but like, you know, there's not gonna be only probably one person working on one question. Like, you both are gonna be working probably at the same time on them, or like discussing, talking, correcting, changing.
Like I won't do that much on my own unless we specify that we would do that. You know. We just saw each other and we were like, "Yo, let's work on this at home," right? Just read through it. That's about the only thing that I agreed with Lisa on. Just read through it so we get a bit of a read. Lisa.
Next to the one thing next to yesterday. That's her name is Lisa.
This is another girl. Lisa's Dutch.
Yeah, I know. I thought Angel was like, I'm so confused.
No.
I don't know who anybody is on this.
Apparently there's like two angels.
Yes, one of them is my the one in my team. I was gonna say my angel.
My angel.
No idea who it is, to be honest. Yeah, isn't that the one?
That's Lisa.
Yeah, that's Lisa. Yeah, I thought that was Angel. They do the same thing. I don't know. Man.
No.
Is that offensive to say?
I mean, people can look alike without being necessarily offensive, right? I'm like the most Dutch person you can like, or German. Some people say German.
Yeah, I could see it. Honestly, it could be Canadian too.
Canadian?
Yeah.
Maybe it's because of the beard.
Yeah.
Yeah, okay. That's me look like this lumberjack. There's an angel. Yeah.
I mean,
Yeah, no, I'm with the first one. Oh, okay, yeah, Angela. Yeah, it's a turtle. I'm with the turtle. You remind me a bit, but not, I guess not that much. But I don't know, there's something about it that reminds me. No, but like, it's not, but like, it's something about like, I guess, yeah, you look, I swear, you look the same, like, especially with the eyes, probably the color. Because, from the fourth.
yeah, but like it's actually at the end. And I would say it's like ice left. Yeah. Almost there. And then
That's a beard too, but I don't know, that also reminds me.
that's my
For what?
Yeah, they said
Is recording working?
It seems to be on, yeah. Yeah, so
there we go.
Oh, yeah.
Or
They shall give them back. Are you gonna go there?
like we'll probably be like half an hour.
You gonna be like, I'm curious about it.
No, just you know what they have to say, and also
The crazy part about everything is that right now the students that are more fun are like questions
also not necessarily because the ones kind of scared for their family.
Yeah, no, yeah, but like at the same time, some of them they have no more money. Yeah,
But apparently apparently it's not what it's called. Also going to go and they set up a group for me kind of thing. Where it needs to go. Subject.
Yeah. No. Like, yeah.
Oh, is that the one?
Yeah.
Also, guys.
I think people like use text names. One of my friends that you're right, no, one of I knew she was like, I really want to give my child a name just in case you want to get just
Wow.
okay.
It's actually a worry I can kind of believe.
Yeah.
Because so let's not necessarily it's really still not done.
As you've maybe noticed, my laptop broke down last week. It's in the repair shop. So I'm getting it fixed, but it's been a bit hard to do work just on the iPad. I borrowed someone's laptop, but it turns out they gave me the wrong password, so I can't use that either. So I guess we're sort of stuck with a brief presentation on neuroanatomy. I had a demo planned on my own brain to just run through it, but I need an app on a laptop for that. So I'm going to try and sort of supplement that part of this with an online viewer that I've tried to set up. So we might get a bit further on that, but as soon as I have my laptop again, I'll record myself browsing through my own brain just to give you a bit of highlight. So the idea is that today I'll give you a bit of background on neuroscience basics, right? And how we pick up neuroscience basics using MRI. And then I'll try to refer back to anything I say during that recording on me going through this brain app thing. As I record that. And I will do that as soon as I have my laptop. Yeah.
Are the slides already okay?
I don't know. So I haven't been able to export it to PDF, but I will do so ASAP. So I apologize for the shit show. It's I don't know. Projects are assigned, right? I have been able to, for I think 85% or something, been able to give you your first choice, but there was an over inscription, or what do you call that, on the somatotopic mapper. So I've had to take some of you who've chosen the second as a second choice, the PRF mapper to the PRF mapper. If you have any questions about specifically that, come and see me after class. Exam date still up in the air. There is a discussion between the rostering and the exam commission. It's not clear whether they can schedule sort of digital exam, proctor IO'd, both here and that you can do it at home. That would be the preferred option now, that it's the original date and time, but it would be, it's your choice of doing it here or at home. And the problem that that would solve is that those of you that have an exam directly after the brain imaging exam, you'd be able to take that on campus, and then you don't have to travel from home to here. Yeah. So that's the solution that they're proposing, and they're trying to see whether they can make that work. But that hasn't been solidified yet. Yeah.
Why not on Friday?
So there's to change the date of the exam at this point in the course is going to be problematic for those students that are, for example, planning on traveling on the Wednesday, for example, right? So you can imagine that there are also a bunch of students that had actually booked tickets for a holiday and can now all of a sudden not do the exam, right? So yeah, we're sort of stuck in between these two situations with that. So I appreciate your patience. Yeah, I'm calling them later today to be able to give you more info. Yeah. Similarly, I was working on finalizing the notebook on MR physics and image reconstruction. That sort of got delayed also because of the laptop situation right now. I want to emphasize that on Wednesday next week, you'll get a lecture by Nikos, my colleague, and I'm our physicist. And the fourth notebook on exactly these topics is meant to supplement that lecture, and the content of the notebook will not be exam material. The content of the lecture will. Yeah.
Still talking about practical.
Yes, this is practical notebook 4 that I haven't been able to upload right now. Yeah. So there's that. I think that I've sort of ticked all my my list of things that I definitely needed to say. Any questions about the state of the course right now? You all have been assigned projects. I hope you're not crying. Yeah, and I do hope that you have a lot of fun. This doing the project, at least in the past, has been a nice thing to be doing. Like, it's a very practical, sort of hands-on thing. Yeah, and also if you have any questions about the content of the projects, right? If you're running into anything, you have until the end of the month to do this, like the week after the exam. We're going to start grading 2 weeks after the exam, something like that. So you won't have to grade each other's projects. So that's taken out also now. You have almost a month to do these projects, right? So what that means is start early. Yeah. Make sure that you know where you're going and don't tolerate being stuck for more than a few days, right? If you're banging your head on a certain problem that you're not fixing, you can always ask us. Yeah. So make sure that you don't run out of time at the end. You should have plenty of time. Yeah. Me being on my laptop, or sorry, on my iPad also means I don't see the chat right now because I can't, share the screen in that way. If there's any one of you that could keep like the chat open, if there's any question, just raise your hand and pretend that you're the person asking the question. Could you do that?
Yeah.
Excellent, thanks.
Someone asked when the project deadline is. I guess exactly.
Yeah, so exactly. So that would be the week after the exam, but we don't know when the exam is yet. So yeah. But so like, what is that? Friday, the 8th of April or something? That would be the day. Yeah. I'll put that in the syllabus also. Yeah.
Is it now shorter exam or is it still discussion?
So there's still a discussion about that. So
It can still be on fire.
yes, it's up in the air. I'm very sorry. Yeah. Yeah. So especially for those of you coming from AI, and then, for example, those of you that have like a computer science background, I think I've received a comment that sometimes you feel like you're being thrown into the deep end. right? With all of this neuro stuff that's happening. And so what I want to do in this lecture is to give you a background on what I think is important to know to be able to find your way around the brain. Right? That will help you also in before doing the project, for example. So the specific things that I'll be saying today aren't necessarily exam materials, right? I'm not going to ask you these neuro things in the exam because this is a brain imaging course and not a neuroscience course. But it will, in many cases, help you understand the brain imaging content by knowing all of these things. Yeah. Was there a question?
There were two comments. One is it possible to have a recording of the third practical?
Yes.
And two, some people are having difficulty hearing you.
Hearing me. Oh, then I'll sit. Is this better? Is this better?
Yes.
Great. I'll sit here. Okay. Can you all see this? It's not that very bright, but yeah, I'll also take these down further. Maybe that room in the new building was really nice with the dimmed lights for the movie and all that. That was great. How about this? Now you'll fall asleep, I know this, but you know, that's fine. Okay. I tested negative, by the way, so don't worry. And it was a PCR test, so you know, I hope you're safe, or that you feel safe. So a bit about neuro background, and I want to go into neuroanatomy also. So what I've done is I've tried to highlight a few of the things that I think are interesting and necessary background. And I'm going to end by just highlighting different aspects of the human brain anatomy that are important to understand and orient yourself in the brain. This doesn't work because it's an iPad, of course. Yeah. So the goal here is to give you enough background to interpret fMRI findings, right? So we were talking about this in the GLM part of the course, right? When you learn how the basic analysis of fMRI data works, you get an idea of how to interpret fMRI findings in that you know that it's a blood response and that you know that you're not looking at neural activity per se and that sort of thing, right? So all of these caveats. But another necessary ingredient for interpretation of these types of findings is always, okay, but what does it mean that there is a blob here and not there, right? And how do that really aids your interpretation. And so what I want to achieve by this lecture is to give you an idea of what different types of functions are distributed throughout the brain. So if I pinpoint some part of the brain, you'll have an idea of what type of function is there, and so forth and so forth. Yeah. Now, I have to also highlight that this idea of being able to point at a specific locus in the brain and then say, it does this specifically, we've already talked about that, right? And this is also mentioned in the YouTube videos that you've gone through. That really doesn't work this way, right? Especially cognitive concepts that we might be interested in are distributed throughout the brain. And so you cannot point to a specific location and say it does this and this only, right? That would be this phrenology that we don't want to be doing. Okay, so what I'm going to do is highlight a few so the sort of components that the brain is made up from, going from small to large. I'm not zooming in completely, but yeah. So we're going to start with neurons and then go stay at the microscopic level a bit. And then I'll start to build everything up into larger components. So we're going from small to large, like it says, right? So what you see here is a neuron. This neuron is what in neural network models, the units are modeled after. Yeah. So if you look at a convolutional neural network, the units there, they're pretending to be neurons like this, but they're much simpler. Yeah. So a biological neuron has an input side and an output side, and it receives information by incoming information hits these synapses on the dendrite. So these are the contact points, these synapses, and they integrate information on the dendritic tree, as it's called. Now, people have tried to sort of model the complexity of the dendritic tree of neurons in the human brain, and they are already performing like functionality on the order of what an entire convolutional neural network does, like a model convolutional neural network, right? So there is a lot of processing already happening in the dendritic tree of a single neuron. Now, what does a dendritic tree do? It takes all of these inputs coming in, all of these action potentials that are coming in, and based on its structure and the timing of these inputs, it decides whether it's going to fire an action potential of its own. Right? So there's an accumulation of electrical signals. It's an extremely complex dynamical process that happens inside every single cell. And then at some point, the cell may decide to fire an action potential. What happens, so I'm not going into the like physics of the membrane that does this, but the conductances of the membrane change very drastically. And what happens is an electrical impulse starts around this part of the cell. So there's integration happening here. This decision to fire happens around here-ish, and then this electrical signal travels along the axon and thereby activates these synapses on the next cell. Yeah. So what we have here is this information flow through neurons, and this holds for all biological neurons, more or less, that you have information flow from input side to output side through the cell like so. Now, depending on the specific function of a neuron, the shape will be different. So different brain regions have completely differently shaped neurons. And that has to do with the fact that there are different requirements on both the axon, so that is the output that the neuron gives, or how it actually integrates its inputs. Yeah. So you will see completely different shapes of neurons, but this general building plan of input on the dendrites and output through the axon is always the same. Yeah? And I just want to highlight that the complexity of what happens in a single neuron, we have no idea of how it works, right? So we have an idea of the basic physics of what happens in these dendrites, but the entire dynamic process that is actually the computation that a single neuron performs is an incredibly complicated process, and we have not very deep understanding of the types of functions that are in there already. Yeah. So what that means also is that if you're in AI and you're looking at like a convolutional neural network that's modeled after the visual system, there's an entire sort of dimension of functionality that this type of modeling is not taking into account. Yeah? Let alone the fact that all of this is a biological machine that's rebuilding itself all the time and is restructuring itself all the time, and so forth, right? So it's much more dynamic also. So what happens is that your brain, people always talk about neurons, yeah, and oh, your brain has this many billion neurons, blah, blah. And this makes it seem as if the brain is composed of neurons, but it's not. By weight, definitely only a minority of the weight of the brain is neurons. Much larger part of the weight of the brain is actually the supporting materials that allow these neurons to function. So there's a whole classes of different types of cells in which, in which these neurons are embedded. You can think of a neuron as performing the incredibly complex and energy demanding computations that the brain is sort of known for, but it needs a support architecture around it, right? So think of this as the Formula One car and its driver, and this is the team that makes sure that it has fuel and that the tires are changed, right? And you see that there are many, many different types of these glial cells that surround the neurons, and each of these have different functions. So there's this sort of paradigmatic sort of idea that neurons do all the communication and the glial cells are just support. But people are more and more realizing also that the glial cells perform communication also, right? The support structure needs to communicate internally and with the neurons also. Just to highlight a few, right? So these astrocytes are called astrocytes because they're star-shaped, right? Astro, yay, star. And site means cell, so there you are, cell. So then you have Schwann cells and ependymal cells. So this is these are cells that are part of the fleeces surrounding the brain. So the brain is actually separated from the rest of the brain by what's called the blood-brain barrier, and the glial cells make up this blood-brain barrier. Just one other type of cell that I think is important to highlight here, because we're going to get back to it later on. These Schwann cells are tiny cells that are around the axons of neurons. And their only role is to produce myelin, which is a protein. And what that protein does is it incorporates into the cell membrane of the neuron. And if a neuron has a lot of Schwann cells around its axon, it can actually be much faster in its action potential. So the action potentials of a heavily myelinated axon, that is an axon surrounded by Schwann cells, those action potentials will travel faster than if not. Now, we've talked about this a bit earlier, in that we can actually find this type of structure, this sort of histology, this character, that's sort of property of the tissue using MRI. So one thing that we're very interested in terms of fMRI is this blood flow, right? And I showed you already this idea that the whole brain is filled with these tiny, tiny vessels. There, of course, yeah.
Schwann cells are easy put myelin.
Yeah, they produce the myelin that allow the axon to be faster in terms of its communication. Yes, thanks. And so, of course, when you are a brain, you need to get blood in, right? And you need to get blood out, right? Because you extract oxygen and things like glucose from the blood. As neurons fire, for example, they need a lot of energy. And so the blood is supplied through arteries and is removed through veins. But it's really in the interplay between these two systems, the supply and the removal systems, that all the interesting stuff happens for us if we're interested in fMRI. So you start out getting blood from a large artery, but that branches off into smaller and smaller arterioles, as they're called. And then in the gray matter of the brain, where all of the neurons are, and I'll get back to that in a bit, you have all of these capillaries, and they are tiny, right? So that the blood vessels are really on the same scale as around the neurons, right? So they're very, very tiny capillaries. So this is where all of the oxygen exchange, for example, takes place. And this entire sort of tree is very dynamic, right? As the neurons that are in this region, for example, start to fire, they tell the glial cells around themselves that it's time for oxygen to be delivered, for example. And this capillary bed then reacts by expanding, for example. And then, of course, you can imagine that if you expand the volume of this here, you'll get more inflow also, right? And all of this is the basis underlying that hemodynamic response function that we are interested in, that we're using in fMRI. Yeah.
So is the concentration of cells or the energy concentration the highest in the middle?
Yes. Yes.
So you have less cells on the left and on the right? Or?
Yeah. So you have to imagine that, and this is shown a bit here on the left. What you see here is the artery, right, and the vein, right? Red and blue is really like the color of the blood, right? So you have cherry red blood is oxygen rich, and darker red, more bluish blood is oxygen low, right? And what you see is that you get, so this capillary bed actually is in the gray matter, right? So the larger veins and arteries are on top. They're far removed from where the cells are, which is in the gray matter. and this capillary bed is where all the neurons are. Yeah.
So oxygen distribution for the brain cell happens in the capillary bed?
Yes. Yeah. So these tiny vessels are much more permeable to oxygen, right? For so this capillary bed allows the red blood cells to deliver their oxygen to the surrounding tissue. And of course, then let's say you have a bold response, right? That means that these capillaries are dilated, right? You get an inflow of this blood, but then you need to, it needs to go somewhere. And that means that it gets taken up by this venules, these smaller veins, if you will, and that then pulls into larger and larger veins on the way back to the heart and the lungs. Yeah. Yeah, so that's really the basics. I'm going to try to show you a bit of the distribution of these large veins in the brain, but I forget if I managed to pick up a slide for this. But so this is just a patch of the brain on the top somewhere, right? And around the brain, there's this web of arteries and veins. Fair enough. The arteries actually, and you can feel this if you just feel on your neck, right? You can feel if someone's alive or not. I hope you've never felt a dead person, but you can feel your own heartbeat really next to your throat, right in your neck. And what this means is that your heart pumps blood through the aortic arc, and then you get these two carotid arteries moving up into the base of the brain. So that's really where the arteries start in the brain, and that's also why we have a lot of pulsation artifacts in the base of the brain. That's because really the base of the brain has these large arteries in there. They're really close to the heart, actually. And so that means that with every heartbeat, the base of the brain really gets this massive pump of blood, which makes imaging those brain regions, even if they are very interesting for functional MRI, very hard, because they're going to be, you're just going to be looking at its movement, more or less. Yeah. Okay, so just to be clear, right? Basic. If we look at an MR image, right, if we take an axial section, in this case of the brain, what we have is this very clear difference between gray matter and white matter. Depending on the contrast of the MR image that we use, that we're looking at, gray matter will either be gray or white matter will be lighter or darker. Right? So the way that the MR image shows us the white matter and the gray matter can be very different. But if you were to cut open my brain, for example, the gray matter would be gray pinkish, and the white matter would actually be light gray. So the gray and white matter, actually, the concepts, the nomenclature comes from post-mortem, just looking at what a brain looks like if you cut someone's brain open. Now, what's the difference in terms of tissue type? Yeah.
Did you say actual cut? This is a coronal cut, right?
Did I say actual? Yeah. Oh, sorry. Yes, this is a coronal cut. I'm very sorry. This is a coronal cut. There's going to be sagittal cuts later. I'm confused. I apologize. This is a coronal cut. So you see one again here, right? Gray matter, white matter. And it's important to realize that the gray matter hosts all the cell bodies. All of the capillary bed is there. The gray matter is where everything happens. It's where the magic happens. And the white matter, on the other hand, is where all the axons are. And it's white because, especially on certain MR images, because this myelin protein is very fatty, and it has a very different composition, this type of tissue, from the gray matter. Okay. Now, of course, what this also means is that if you are doing fMRI and you're looking at where the activations are, where the signals are, you'll find hardly any in white matter, and all of them will be in gray matter. So that's where the oxygen is being consumed. That's where the capillary bed is. And that's also where the axon and the cell body of the neurons is. Now, you also have to realize, and this is more or less an aside, but the neurons in the gray matter usually just communicate very locally. It's not as if all of the neurons have axons that go through the white matter. The white matter, these bundles of fibers, are really for long distance communication, and most of the communication in the brain is very, very local.
The connection and also using those accents and rights
Yes,
within the gray matter.
yes, yeah. So the length of axons can vary dramatically, right? So for many of these cells in the gray matter, they are communicating locally, and their axons will be, maximally a millimeter long, right? But neurons in the motor cortex actually have their terminals all the way down in the spine, so they can be more than a meter long if they're innervating the legs, for example, right, all the way down into the spine. So that's very, very, very valuable.
So the cell bodies are only found in the gray matter.
Yes, yes. So that's the neuron cell bodies, right? Because all of this myelin, of course, is these Schwann cells. So there are a lot of cells in the white matter. It's not like it's dead or anything. And so there are also vessels in the white matter that supply these Schwann cells with their energy, right? But they don't react to neurons firing. And so that's why we don't measure them using fMRI. Great. Yes, so coronal slice. Sorry about that. It's a beginner's mistake, really. And so this is just to show you a bit of where things are. I'm going to go into a bit more detail because I think it's interesting for certain parts of the brain. What you see here is the side view of the brain, a sagittal view. What you see is this subdivision of the cerebral cortex into lobes. And this figure also highlights a few of the other regions of the brain. And you see that there is a difference in folding, for example, between the cerebellum and the cerebral cortex. Yeah. Now, the cerebral cortex is the newest, what do you call that? It's called neocortex because it's more recently evolved than the rest of the brain. And this, for example, the cerebellum is much older in terms of evolutionary history. And I want to highlight that this is arbitrary. So what we're calling, yeah, go ahead.
What's the neocortex? It's everything above the cerebellum.
Yeah, so anything that has sort of this structure, this entire thing is the neocortex, right? So that is anything that has lobe attached to it in terms of its name is the neocortex. And so you have to imagine that, for example, rats also have a neocortex, but it's much, much smaller. It also has a front part and a back part and all of that, but it's much, much smaller, and it's much less folded than it is for us. So what happened in terms of evolution is that our function required all these mental faculties. We needed brain tissue for this in the neocortex. And what happened is this grew from a sort of flat sheet into something that had to be crumpled up inside the skull. Okay? So I think I said this before, right? It's as if you take a tablecloth and you just push it into a fishbowl. If you look into the fishbowl, then you'll see something similar to this. And that's really the type of, yeah.
And just another question with terminology. Cortex is in everything, or is cortex the same as cerebellum?
No, so yeah, so cortex means bark. It's just a Latin word for the bark of a tree, right? So it's everything that's on the outside. And so we have this neocortex. There's also an older cortex. I'll talk about that in a bit. But yeah, so you can definitely also tell this from the way that it looks. The neocortex is, in humans at least, characterized by the fact that it has these gyrifications that I was talking about, right? So that's the folding at this scale. We'll go into that in a bit some more. So I want to highlight that the presence of these lobes is also pretty old evolutionarily, but it's also a bit arbitrary because it's one big sheet, the neocortex. The reason that we're calling it lobes is that these sulci, that is anything that goes in is a sulcus and anything that comes out is a gyrus. So those are two terms I won't quiz you about, but that's what you can. So anything in this, these are just a bit deeper, these sulci, sulcus, sulci, Latin plural. These are just a bit deeper, and so people say, there's different lobes. And these are sort of separators, but you shouldn't take this too literally. But
this is the general way in which we divide things up. We have 4 lobes of the neocortex: frontal, parietal, occipital, and temporal. Yeah. Let me just go through what the canonical features of each of these regions are. Right? So the temporal lobe, it houses the auditory system at the top, and it houses things that have to do with image recognition and sort of episodic knowledge at the bottom. So there's this auditory, and then the bottom is visual. Yeah, so if you're coming from AI and you've looked at like ResNet for vision, right, for image classification, that architecture of a convolutional neural network really is made to resemble this bottom of the temporal lobe. It's the part of the brain that we do image recognition with. So let's go further, like the frontal lobe. The frontal lobe is, and I'll talk about the frontal lobe a bit more because it's a beautiful part of the brain that we understand very little of compared to other parts of the brain. This really does things like the development of behavior, things like planning, movement, anything that's oriented towards the outside world, in either now or in the future, is the frontal lobe. That's really what the frontal lobe has evolved to do. Moreover, the frontal lobe is the lobe of the 4 lobes that we're talking about that has increased in size most dramatically, going from our ancestors to humans, right? And you can imagine why that is. We as social animals, as planners, require a lot of brain tissue devoted to things like, but if this person does that, and then that might mean that in 2 weeks there might be this, and then that means that I should be doing that in a month. That type of reasoning is exactly what frontal cortex has evolved to do. And I'll get into that a bit more. The occipital lobe is where the visual information enters the brain. And because we are such visual animals, it more or less only does vision, the occipital lobe. Yeah? And then you see the influences of vision moving into the temporal lobe and into the parietal lobe. The parietal lobe is beautiful because it houses the somatosensory cortex. So that is the parts of the brain that do the feeling of your body, right? So I now feel that my right arm is extended. Not only have I done that, but I also feel that that is the case. And that feeling is actually the part of the function of the parietal lobe. Moreover, the parietal lobe actually brings together a lot of this sensation of where am I in space, and for example, the visual information that goes with it. So it's a very complicated piece of cortex because it brings together many, many different functions. And you can also, so here we see language being a very prominent aspect of parietal lobe. You can imagine that your words are more or less arbitrary labels on abstract things, right? And this abstraction is really what the parietal lobe evolved to do. Right? It doesn't care whether something is being felt or seen. It has a representation of that object, regardless of whether it's being felt or seen. Right? And you can imagine that is one stepping stone towards using abstract labels in terms of language. Now, the neocortex, people overemphasize its importance. There are patients, for example, that went to the doctor when they were like 40 and said, yeah, I have this headache and I have trouble standing up sometimes. Can you look at my brain? And then they do this brain scan and it turns out that they only have 20% of their neocortex, very thin, and it's just, all over the place. You can get around with large parts not there if you grew up in that state. So like the way that humans think that they're the pinnacle of evolution, right? Similarly, humans tend to think, or researchers tend to think, that this neocortex is all that we should be caring about. But that's definitely not the case. And I'll also argue that as we speak about this. Now, 2 things that I've not talked about here is the brainstem, which it's close to the body, as you see, right? And it's actually large parts of it are mostly an elongation of the spinal cord, right? So you definitely see that this is the part of the brain that connects to the body. It houses very basic movement functionality, things like breathing, things like heart rate regulation, temperature regulation, hormonal interface between the body and the brain. All of this happens in the brainstem. It is beautiful. And then there's the cerebellum, which is this really weird cousin of the cerebral cortex, the neocortex. It's just underneath there. People have been disregarding it a lot. It's been there for ages in terms of evolutionary history. And people thought that its role was just to coordinate movement. So if your motor cortex, which is here, sends a signal down your spine to move your arm, for example, there's a lot of processing that happens between your motor cortex and the cerebellum back and forth to optimize, for example, the trajectory of your hand as you're moving. And so people have thought that cerebellum is important for things like balance and coordination, and it's just motor. It just does motor. So it's involved in things like timing. It's very timing specific. We're finding more and more that the cerebellum doesn't just do motor, doesn't just do action, but is involved in more or less all types of cognition, because this coordination, and specifically coordination in time that the cerebellum does, is important for all domains of cognition. I also want to highlight that this sort of broad functional localization, sight, for example, for the occipital lobe, is a caricature. Right? It's the first lesson that you teach, that you get about how the brain works. But if you really dive into this, you'll find that there is a visual region here in the frontal lobe. It does things with eye movements, but it's definitely a visual region also. Right? So it has this interface between vision and action. So calling this only action really isn't correct. And similarly, if I move my hand in the dark, there will be regions in the occipital lobe that light up because they are generally used for seeing hands, for finding out what hands are doing. And so there's a lot of communication, and all of these functions are much more broadly distributed throughout the brain. Yeah, so, yeah.
Yeah, I don't know, and I make a mistake. You said before that the temporal lobe, the bottom of temporal lobe, which related to the image classification, is that correct?
Yes.
But it seems like it's more related to auditory part of the function, and the occipital lobe seems to be related to the side.
Yeah. No, so I want to reiterate, this is a caricature, right?
Yeah.
And the temporal lobe is very complex. So let me just go into that for a bit because it's interesting. So this upper bank here is a superior temporal gyrus, right? The gyrus is this sort of sausage type thing where the cortex bulges out, right? So the superior temporal gyrus here, that is is auditory, right? And the auditory signals come in here and then they distribute throughout this auditory cortex. And so, for example, you start out with selectivity in the neurons for pure tones, right? So is a tone high or is it low? But then a few synapses further, a few processing steps further, you have regions that are very specifically tuned to voices and to speech, right? And it turns out that when you look at the bottom then of the temporal lobe, there are regions that are very selective for processing faces. And especially those regions that are face selective in the superior temporal, in the temporal lobe, visually are very close to the speech selective regions in the auditory parts of the temporal lobe, right? Because when you look at me, you hear me speak, right? And what you see my face doing actually helps you interpret what I'm saying auditorily, right? And so that's really where this interface between both audition and vision happens in the temporal lobe. And then, for us, that's really important, right? Because we're social animals. We need to understand what the person is saying, right? And what they're intending by what they're saying. Does that clarify things? Yeah, cool. Sorry if yeah, go ahead.
I've got like a general question, but like how was this clear, like that each area of the brain was for this function? Like it's fMRI before that? Or like
how people started with this, yeah. So you may have heard of Broca's area in the brain or Wernicke's area in the brain. Do those things sound familiar? No? That's not important. They are both language-related regions, and Broca's area in the brain is is responsible for the production of speech, and Wernicke's area is important for the comprehension of speech. Right, so they're like the active and passive parts of speech. Broca's area is somewhere around here, and Wernicke's area is somewhere around here. Why are these called like this? These are weird names. It turns out that Broca was a French neurologist that had patients that stopped being able to speak. Then after they died, he cut open their heads and he found that exactly this region of the brain had housed a tumor or had experienced a bleeding or a cerebrovascular attack, had experienced damage, right? Similarly, Wernicke was a German neurologist that found exactly the same thing, but then, by accident, had this group of patients that had the bleeding here and thereby produced these symptoms of not being able to understand what people were saying anymore, yeah? So this idea of functional localization really stems from this time frame in like the late, like the 1860s or something. I think these were discovered. So that's when people started to realize, hey, there's a specific function attached to this region of the brain. And that's the inheritance of that really is that we try to look at blobs in the brain and say, this does that, right? But it's important to realize that when you have a lesion somewhere in your brain, all of the other brain regions that are attached, like that communicate habitually with that region, will also experience problems, right? So it's this idea of function being easily pinpointed to a specific location. Yeah, there's a lot of problems with that. Does that answer your question?
Yeah, so we are sure about like different function of the brain, but not too sure. Like also like how it is described here. Like
yeah. So I think the general gist is that we started out thinking that each specific brain region had a specific function, right? Like the Broca and Wernicke areas. And we are finding more and more that for the types of complex cognition that we perform as humans, we need much more integration, right? And that means that functionality is distributed throughout the brain.
Yeah. Is it a random question? But someone asked if it's possible to open the camera.
Open the camera, like for looking at me.
I guess.
For looking at the screen, I hope. Yeah. I'll check because I think I should be stop video. It's on now, right? Yeah. Let me and I'll now switch to the presentation. Is it still on or did it just shut off? Cannot see? Yeah, I'm sorry. Then it's the iPad thing. I'm very sorry. But, I don't like looking at myself, neither should you. So then I just left out a huge part of the brain is so complicated that we could be here for hours. I just left out a huge part of the brain. I talked about the brainstem and the cerebellum and the neocortex, right? And it turns out that when you take out the neocortex, you can actually look at much more of the brain because it actually encapsulates beautiful structures, beautiful brain structures that are somewhere between the brainstem and the neocortex. And so what I've done here is I've tried to give you a little idea of what's what. Again, this is not this is not exam material, but there are these beautiful divisions of labor in the brain. And I just want to highlight a few of them. So we haven't really looked at the parts of the subcortex, so that's everything below the cerebral cortex, that isn't brainstem, and there's actually quite a lot. And you have to think of these brain regions as having evolved in the reptilian stage, right? So these are about 100 million years old evolutionarily, whereas the neocortex is younger, right? And it's a more mammal-like structure of the brain. And so our brain is really like stacked levels of evolution on top of one another. And what that means is that we've evolved neocortex to be larger. That neocortex didn't just function alone. It has evolved to cooperate with these more primitive regions of the brain, right? That also means that the function of a frontal lobe region is going to be highly connected to what these regions are doing. Now, there's a few subdivisions that I want to highlight. There's a whole group of regions that are called the basal ganglia, and they are incredibly important. What I've done here is I've highlighted everything that is part of the basal ganglia in blue. And so you see that it really is this smorgasbord of different regions. So this subcortex really is this beautiful puzzle of separate regions in all these weird egg-shaped type regions in the center of the brain that's surrounded by this neocortex. Now, the basal ganglia as a system evolved more or less around the same time, and it has this beautiful functionality. And usually what people do is they summarize this functionality as, do I stay or should I go? Should I stay or should I go? And so you can imagine that in an animal's environment, answering this question all the time is key to survival, right? If you if you decide to go and start walking when you shouldn't, right, you get eaten by a lion or whatever, right? If you decide to stay when you should be acting, you end up hungry because you didn't manage to gather your food, for example. Yeah. So there is a lot that you need to do as an organism to organize your behavior exactly in these terms, right? So things like, do I act? Should I inhibit my actions? How rewarding is this specific action that I might perform for me? So these basal ganglia regions all do different things, and there's beautiful mathematical models of how these regions interact to deliver us our behavior. And the basal ganglia are also specifically broken in many different disorders. So I'll get into that in a bit, but they are beautifully responsible for these basic ideas of want and not want. And then even like lower, there's this set of regions called the midbrain that are very important in things like emotion regulation, and again in things like reward processing and fear processing, that sort of thing. And then there's a few regions in the brain, the amygdala and the hippocampus being examples of those, as you see here, that are very involved in memory, things like episodic memory, what happened to me when, right? That sort of thing. Things like navigation and emotional memory. So you already see that, for example, if you once almost got eaten by a tiger, right? What does your brain have to do to recognize a tiger and make sure that you don't walk in front of that tiger the next time? Well, you need to recognize the tiger, right? So you need visual processing in the temporal lobe, in the case of the human brain. Then that needs to connect to the amygdala, which does fearful memories, which is responsible for fearful memories. That then informs your basal ganglia to inhibit any action that has to do with running in front of the lion. Or was it tiger? I just said, right? Coronal, axial, whatever. So you already see that this type of functionality in a natural situation needs to blend all the time. Nonetheless, there are these specific functional responsibilities. And then finally, we have the thalamus, which is really at the center of the brain. And it serves as a relay station. So any communication that comes from the sensations, so from, let's say, audition, vision, somatosensations, so touch, but also motor things, all of those travel through the thalamus to the cortex, and then when the cortex is done with its processing, it also sends things back through the thalamus. So it really is this sort of switchboard for signals traveling to and from the cortex. When I say cortex, I mean cerebral cortex, the neocortex on the outside of the brain. So the thalamus has all of these small parts, and they are all responsible for different things. So you can find an auditory region in the thalamus. You can find three visual regions in the thalamus. You can find motor regions in the thalamus, and so forth and so forth. It's a beautiful structure. Whole books. I have a book about the thalamus. It's 1,500 pages, and it's already old. So yeah, all of this is incredibly complicated. Yeah. Are we up for a break already, or should I just keep talking? Mr. here is nodding. Yeah, we're up for a break. Yeah, let's have a break. Be back in 10. I could talk until 8, like late.
There was also another question I didn't answer earlier. I wrote it down. That's what I copied it. I've answered them in the chat. Or you can just say.
Oh yeah. Yeah, so the question is concerning Perusol. Its grading functionalities sort of holds or becomes quiet after a deadline. I am not going to take any of that into account, right? So you should be able to still interact at least with the PDFs, right? I'll check this, but yeah, it is still recording what you're doing, so you should be okay. Yeah, and again, there is no strict grading of anything that you're doing.
I have some like interesting questions.
Yeah, tell me.
Yeah, just related to this course.
Someone said so you still get a bonus if you do it after the deadline, but there's no bonus.
Yeah, so the bonus is part of me looking at what you've done, and what you've done is still being recorded after the deadline. Yeah. I think I have to like find a way to pause the
recording in progress.
Yeah, I see. You both are the same.
Next to the deadline.
So that course is mandatory course, or you can select it?
No, you can select the other ones. The registration.
Yeah, I didn't want to like register for.
Oh, you are trying program. So I know you are trying program.
Okay, let's start it back up. That was such a refreshing break, wasn't it?
Yeah.
Yeah, it feels so much better now. So what I want to do now is give you an example of, we looked at these lobes just now, right? Okay, a lobe. The lobe is if you were to iron that part of the cortex out, you would end up with like a patch, like this big, right? A flat patch this big. Of course, it doesn't do one thing. There's a lot of functionality that's distributed across such a patch of cortex, patch of the brain. And I just want to use one of those lobes as an example of how function is in general distributed through one of those lobes, and also how, in this case, the frontal lobe then interacts with other parts of the brain to perform those different types of functionality. Now, one thing that we think happens in the frontal lobe, it's very hard to understand exactly what it's doing, because what it's doing is so abstract. We really don't have a simple model of the types of functionality that it performs. But we know a few of these sort of basic building block type things that are, I always think, really, really beautiful. And so if you look at the frontal lobe, you first of all, you see that it's huge. It's a very large part of the brain. If you then start looking at it, there's a beautiful study from 1937 by a neurologist, a neurosurgeon called Wilder Penfield. He won the Nobel Prize for this work. So he would operate on epilepsy patients. I think I talked a bit about this before, but what they did then was to like open up the skull. I really take like a fist-sized chunk of the skull, like saw it out, take it out, and then look at the brain. And what they would do is they needed to sort of know what part of the brain did what, because they're going to resect parts of the brain to solve the problem of the epilepsy, to cure the epilepsy. Okay? So epilepsy is this problem in the balance between inhibition and excitation in the brain. Inhibition is to clamps down activation, and excitation makes activation larger. So you get these sort of electrical storms, and usually they originate in a specific location. So you want to take that out. As a surgeon, you just want to take a little spoon and spoon and like take it out, right? This little origin of the epilepsy. And then the epilepsy is solved. And that actually can happen. Like in some patients, when the surgeon is doing their job well, they end up just having no epilepsy afterwards. But you need to find where that is, and you need to know exactly what parts of the brain you can resect and which you cannot. So what Wilder Penfield did is he took this electrode, a stimulation electrode, and would sort of zap specific parts of the brain. And he would find then that if you zap a part of the brain around here, you get like hand twitches. And if you zap a part of the brain here, you get more or less like shoulder twitches. You zap a part of the brain here, you get facial twitches, or people go like, huh, or something like that. And he found this beautiful structure already there. So our knowledge of this layout of the frontal cortex started there, in that it does action, right? If you zap a specific part of the brain, a specific part of the body will start moving. But there's another gradient I'm always in awe of. So the primary motor cortex really drives single movements, like for example, of a finger. So if you have a cell there, it's responsible for moving this part of my finger like so, right? That's what one of those cells would be doing. And if you then move from this primary motor cortex to the front of the brain, these action plans, as you might call them, so you can have an action plan about doing this, right? It's a very simple action plan. And that's in the primary motor cortex. But then in the more anterior part of the brain, so that people call premotor cortex, you might have something that is a simple grasp, right? A simple grasp of, for example, this lovely cup of coffee, right? But then, of course, actions need to be planned even further. Is it appropriate that I now grab my coffee cup, right? Maybe it's still too hot and I don't, I'm just going to be holding it, right? So as you go from this primary motor cortex to more anterior regions, it becomes so that the processing is less direct action and more abstract plan. Do I want to, right? Or what would happen if I? That sort of processing becomes more and more dominant as you move towards the front of the brain, what we also call the anterior part of the brain. And then there's the posterior side, and caudal and rostral refer to, rostral stands for nose, and caudal stands for tail. So that's the front and the back in another way. So what you have is that you have direct actions here, and you want to control those actions. And for that, you have this hierarchy in the frontal lobe where the further you go to the front, the more abstract that question of control becomes. And that's really beautiful because it means that things like your personality, right? Whether you are a person that likes to just go for things, or whether you are the type of person that just wants to look and see first, sort of thing, all of that we think is in the frontal cortex, in the frontal lobe. So there's a few other dimensions of organization in the frontal lobe that I want to highlight. One is orthogonal to the one that we just saw, is that if you go from the top of the brain to the side of the brain, from here to here in the motor cortex, what you have there is a very, very beautiful map of the face. So I just already highlighted this, right? If I zap this part of the brain, you have a twitch of the finger, for example, and below here you have the face. Okay. So if I ask you to move your lips, I will get a blob of activation in my fMRI experiment that will show activation here. It's very localized. Okay?
I was going to say, some people are saying that when you say here, they don't see where here is.
Oh yes, I'm sorry.
You can't really say
here. I'll try to stop saying here. I'm sorry. Yes. the last time I said here and pointed at something, I was moving the pointer from the top of the brain on the right side, the PMD, to the PMV. So there's, yeah, that's what I just did. All the others, I can't recall. So what I want to point to here is this beautiful organization of a body map on the left inside this motor cortex. So you can see, you can imagine that very concrete movements need to be tied to the body map very, very concretely, very specifically, whereas more abstract plans are more and more abstracted away also from that body map. Now, you can also imagine that when you move something, not only do you need to perform that action, but in order to function in the world, you also need to anticipate what that action will cause. And I don't know about you, but well, no, in general, actions cause feelings, right? If I do this, I feel that I'm moving my arm and I feel my fingers hit the table. You also can't see that. Yeah, so I was moving my arm and then my fingers hit the table. So a motor action is almost always accompanied by a touch sensation. And what that means is the brain needs a very, very direct communication between bodily actions and bodily sensations. And the brain's organization actually gives rise or sort of embodies this literally. Because if you look at on the right, everything anterior to the central sulcus, so that's on the left in the red, is motor. And right next to it, on the right of it, on the other bank of the central sulcus, what we call it, is touch. So motor and touch are right next to one another in the brain, and the motor map and the sensation map are actually aligned beautifully from the front to the back of the central sulcus. Okay? And this has to do with the fact that we always interact with the world, and when we act, when we perform a bodily movement, we also feel that movement in our touch. So what that also means is that this in the parietal lobe, so that's the blue on the right, you have very concrete bodily sensations right next to the motor regions, and more abstract bodily sensations are further into the parietal lobe, so further to the right. There, for example, they mix more with visual inputs, like where exactly am I moving my hand? If I want to know that, I have to integrate the sense of touch with the sense of vision, for example. So that's one direction in which the frontal regions are also organized. And you can find these body maps everywhere in the brain. So they're everywhere. And that, what I think that shows is how strongly our bodies are, our brains are really in the world, right? So it's not like we're a brain in a vat. Our brain communicates with the world. We exist in the world. And that always happens through sensations, in this case, sensations such as touch. Yeah?
You just go back to previous.
Yeah.
So you have those blue regions in the left and the right hemisphere.
Yes.
Is it then that the function of the limbs are like those are on both sides?
No, they're crossed.
Sorry.
They're crossed. So if the left hemisphere of the brain controls the right side of the body and vice versa.
Yeah. Your nose, for example.
Yeah, it's the left. Yeah, so left side of your nose, right side of your nose, sort of thing. Yeah, yeah. So in general, when you're closer to the midline, such as in the face, it's yeah, the two hemispheres of the cerebral cortex start to cooperate more, and we definitely see that.
Also like the middle of the brain.
No, it's actually on the side. So the face is, so the hands are here. I'm pointing at the top of my head, and then the face regions are on the side of the brain. Yeah.
You said that the left part, right part control the left hand.
Yeah.
So the similar blue face.
Yes. Yes. And it's similar to vision. I don't know if you remember from when we talked about vision.
Yeah.
Vision, the lens flips everything left, right also, right? So this also means that the left hemisphere controls my right hand, but it also controls, it also gets information from the right part of the visual space. So that makes sense, right? The motor map and the visual map are actually aligned in the left-right direction. This is an aside. This is not a rule that things should always cross. For example, the cerebellum also has beautiful body maps. The cerebellum, that little brain on the bottom, right, that does coordination. It is actually not mirrored. So there, the right side of the cerebellum controls the right side of the body. It's completely weird. Here we are, evolution, you know.
But like the tongue, you only have one, so
Yeah, so there's a lot of coordination between the left and the right side of your brain. Yeah. And so there's another division here, which is on the right between the top and the bottom. So the top shows the lateral portion of the brain. So that's on the side of the brain. And that part of the frontal cortex has to do with things that are more externally driven. So that is, do I react to the world, right? That's driven by information coming from the outside. Whereas the medial portion that you see on the bottom there, that's the medial surface of the brain. Those parts of the frontal lobe are more about internally driven things like what do I want? What do I feel like?
The image on the right bottom. Is that what you're talking about?
Yeah, so there's like on the right side of the screen at the bottom, that where it says SMA.
Yeah, the right. So the red one is the internal.
Yeah, where you see the entire brainstem is the center part of the brain. So you're looking at the middle of the brain. Okay, so these are both sagittal sections. It's only so you're looking at the top. You're looking at the side of the left hemisphere from the side. On the bottom there, the smaller one, you're looking at the inside of the right hemisphere.
So it's like this.
Yes, it's like we've taken out the left hemisphere out of the skull, and then you're looking into the brain onto the medial part of the right hemisphere.
Okay, and the blue region is for external.
So the blue is for sensations, the red is for motor actions on the right, and similarly on the left. Yeah.
Okay, so what was then the external versus internal?
That's whether you're looking at the lateral part of the frontal cortex or the medial part of the frontal cortex. Yeah. Great. I'm not quizzing you on any of this. I just want to give you an idea of, the brain is this enormously complex thing, but there are these beautiful sort of choruses of organization, right? There's like different dimensions of organization that we can definitely speak about. Now, I want to quickly, not even quickly, I'm going to go slow. But I do want to highlight this beautiful, we now talked a bit about this architecture of the frontal lobe, this organization of the frontal lobe is actually mirrored very strongly in its interactions with these basal ganglia that we spoke about just now, right? So these basal ganglia are these should I stay or should I go regions. And you can already understand that all of this frontal stuff, right, even direct actions or abstract actions, they have to do with whether you should do things with things like reward and all of that sort of thing. So your brain needs these more primitive and more novel parts of the brain to interact, to give rise to things that have to do with, getting what you want, yeah, and avoiding what you don't want. So there's a lot of computation that has to happen at different time scales, right? So you have this gradient of abstraction, which also is a gradient of direct versus indirect, or like now versus future. Yeah. And that you require a lot of interaction between the frontal lobes and these basal ganglia things. And what we see in the basal ganglia, at the bottom here, this is a little cartoon of this very complex schematic of how these different basal ganglia regions interact. I'm not going to go into that. We could devote a whole, it's beautiful. But important to take away from this is that these regions, they are very, very fundamental to how we move and how we direct ourselves in everyday life. And what that means is that when people suffer from things like addiction or in diseases such as Parkinson's, we see damage to specifically these regions of the brain. So, or dysfunction in the basal ganglia regions of the brain. So I hope you get an idea for this part of the brain being responsible for action, right? And that all the ingredients that have to do with developing action are also, intertwined in these brain regions. Now, in this case, I want to highlight the fact that we have these different neurotransmitters that play an important role in exactly all of these processes in the basal ganglia. And I want to highlight dopamine and noradrenaline that I'm sure all of you heard of. Right? Who of you have heard of dopamine? Great. Did you know that a banana has more dopamine than your brain? If you were to take out the dopamine from a banana and put it in a syringe and be able to inject it into your brain, you'd probably die. But because we have a barrier between the blood and the brain, and our bodies actually buffer all of it, you don't experience a sudden high when you eat a banana. It's a shame. It really is a shame. But yeah, so I want to highlight a bit about sorry, neurotransmission. Just a bit of background. For those of you coming from AI, for example, you sort of have these neural network models, and there a unit is a unit, right? And it's supposed to resemble a neuron, but neurons are way more complicated. And this is one of the ways in which I think neurons are much more complicated. So when a signal from one neuron arrives at another neuron, right? So this is this stream of information, right? So you have input arriving on the dendritic tree. That signal of incoming information can actually be patched through in different ways. So the principal way is for that action potential, that signal, to arrive on the dendrite, and then to change the, that's what we're seeing on the left here, to change the electrical properties of the membrane there, such that the cell that's receiving this message is going to either be more likely to fire or less likely to fire, right? And we call this, so this is direct electrical transmission, right? So when, for example, you see something, it means that the signals from your eye are being patched through directly, from cell to cell in this electrical way. Yeah. So the incoming signals actually directly evoke electrical signals to be generated in the next cell. And this can be done really, really fast. So millisecond timescales. Yeah. So that means that this gives us for example, in our visual system, if I flash a light, that signal will actually travel through the entire visual system in something like 150 milliseconds. Yeah, so that's a whole bunch of different synapses that it crosses, so it's really, really fast. Yeah.
Someone asked, how did they research this using the brain of deceased people?
No, you can actually, so all neurons have these receptors and all of these, and these things like ion channels and things like that. When you, if you, for example, go into a neuroscience class, you get practicals. They use frogs, for example, or snails, or animals such as that, because most of this sort of cellular machinery we actually have completely in common with those types of animals. This is really, really well maintained in terms of evolutionary history. Yes?
The firing of neuron, is it like it fires or it does not fire, or is there like a level of firing very high action potential?
Yeah, so what usually happens is that the signal accumulates in this dendritic tree where the inputs come in, right? And then some threshold is reached where all of a sudden the entire cell goes like, okay, now I'm going for it, and it fires this action potential, which sends the signal out along the axon.
But that, for example, it fires the action potential. Can there also be a level of?
No, it's all or nothing.
It's all.
Yeah, yeah. So there are certain exceptions to this rule, but the 99.9% rule is all or nothing.
But every neuron is a different threshold, right?
Yes, yes. And what's cool is the right part of this, which is the non-directly electrical part, is called the modulatory or metabotropic. these are different types of receptors. They work with different neurotransmitters and they modulate. So what they do is they don't directly try to have an electrical effect on the neuron that's next door. What they try to do is say, like in general, right, you should be firing more. So you should lower your threshold. Or in general, you should be firing less. So you should make your threshold higher, right? So these are modulatory. What this means is that your brain can really change its state by manipulating these things on the right. So the metabolism of cells, how they function, right? So it's at a meta level that these types of things function. And exactly those neurotransmitters that we were just talking about, like dopamine, the reward neuron, right? Noradrenaline, which is like a stress hormone. It's like in anxiety and things like that. It's very important. Or mood, like serotonin is very important in mood. All of those slower things, they're modulatory. They change the way the system works, right? So, yeah.
These are cells that influence those neurons?
Yeah, so there are specific cells that output, for example, dopamine. Now, let me go to the next slide because there I just have a slide for it. Again, good questions are the ones that can be answered by a click on a mouse. So yes, different cells. And specifically for these modulatory neurotransmitters, there are, so these ionotropic electrical neurotransmitters are everywhere. All of the, like, most of the neurons work by just direct electrical transmission. But you have these systems in the brain that use these metabotropic modulatory neurotransmitters to fine-tune the function of all of these electrical signals.
Is dopamine like a neural?
Yes,
So
yes.
it's just the fluid that then is also the dendrites that accept that in the neuron and then they.
Yeah, and they change their behavior
So it's
over.
actually the dopamine itself that changes the situation.
Yes. No, well, so that's what's in here. It's quite so on the right side here, you see that the neurotransmitter hits this receptor on the left, which is the pinkish heart-shaped thing. And then you have these enormous cascades of biological processes inside the cell that change how that cell functions, right? So, for example, through this enormous cascade of biological processes, that neuron might then say, oh, I need to make more electrical receptors, right? So that whenever this electrical signal enters, it's going to have a stronger impact, right? So that these are slow processes that are much more complicated. Yeah, does that answer your question?
Yeah.
You had another?
Yeah, I have a question just in general, like basically if I want to produce those modulatory effects with those dopamine, those hormones.
Modulators, yeah, hormones,
But
yeah.
then the thing is, are the cells that produce those hormones, or like, do they get an electrical signal that says, okay, with that electrical signal, then I will send that message forward to the cells to change the.
Yeah, good question. Let me try to answer this by an example. So on the left here, you see dopamine, right? And I don't know if you've ever heard of the pleasure center of the brain. Well, it uses dopamine, right? So there are specific regions in the midbrain, in that subcortex that we spoke about earlier, where the cells are only dopaminergic. And what that means is that the cell bodies are in this region. They have axons that project elsewhere. Whenever they receive an electrical signal, they will fire, they will send an action potential, but the result of that action potential is not electrical activation at the end of the axon, but dopamine there, right? So whenever these neurons activate, they will start to modulate other parts of the brain. Yeah? And so for dopamine, this is very beautiful. There are actually two semi-separate regions in the midbrain that produce dopamine. One is called VTA and the other is called SN. You can see them there on the right in the noradrenaline figure. They're the red blobs in the center of the brain. So VTA is this pleasure pleasure center. So if you take a rat and you stick an electrode in its VTA and you start zapping it, so you start putting currents into the electrode, you will cause the cells there to fire, and you will get floods of dopamine entering the rest of the brain. And this will feel to the animal as like heroin or orgasm or something like that, right? So it will be extremely pleasurable to the animal, and they will get addicted to this. If you can make a lever in their cage that causes this electrical activation of this VTA, and they will do nothing more than just hit that lever over and over all the time until they die. Right, so I don't know if that went anywhere otherwise, but yeah, so that region is responsible for projecting dopamine to those regions that guide behavior based on reward. Yeah. So you can see here that, for example, this feeling of reward, of pleasure, is something we seek, right? And what that emotion and what its role in cognition is that it guides behavior on a longer term, right? So what that has is it needs this interaction with the frontal lobe, right? And with the basal ganglia that control action. That's what it's supposed to modulate. And this is modulation that should be occurring on a longer time scale, right? If you really like, if you taste peanut butter and you want to have more of it, then, what do? Well, you go to a supermarket and you have to buy peanut butter. That's that's the difficult path of actions, right? But that's the type of thing that type of processing changes. Is that clear? Similarly, for things like stress or arousal, you have similar things, right? If something scares you, you can feel the hairs in your neck rise up, right? It's a hormonal thing. Similarly, that neurotransmitter, noradrenaline, has this stress function in the brain. The adrenal gland, the adrenal gland, so this is the part of the kidney, on top of the kidney, it produces adrenaline, and it has these stress effects in the body. That same neurotransmitter with minor modifications is used as a neurotransmitter in the brain only. And there it also does things like arousal, right? It widens your pupils, it makes you more alert. For example, if you take Ritalin, that actually activates these dopamine and noradrenaline systems so that you become more alert and aware and that sort of thing. Yeah. So this is really the role of neuromodulation as a general principle. I could talk for ages. They interact beautifully and they share biological mechanisms and all of that. It's gorgeous. But is that clear? Any other questions about how this works? Because you can imagine that, I don't know if you remember this, but I talk about this monkey finding where they were able to do electrical recordings from neurons and looking at this bold response in the monkey. Yeah, and we saw there that even when the neurons are silent, so when there is no electrical activation of the neurons in the visual system, when the monkey expects there to be something, expects something to happen, you see this enormous fluctuation of the bold response still, right? So this we then talked about in the context of this indirect relationship between neural firing, the thing that we might be interested in, and the bold response that we measure also with fMRI. Yeah, so there's this disconnect between them. But it's very likely that especially those types of bold response that we measure also using fMRI are caused by things like this, by neuromodulation, right? So it definitely is a factor in brain function, but it doesn't, it doesn't align with electrical activation of the brain directly. It's really the modulation of the brain. And people are more and more understanding that when you're measuring bold responses using fMRI, in humans also, you're measuring this weird mix of the electrical activations, the direct functionality, and this type of neuromodulation. I think I'm almost, I'm done with my slides because my plan was to now show you stuff on my laptop. Yeah. But I can't. So what I'm going to do is get my laptop back and then record myself talking while I go through my brain. Yes? I'm going to, in a separate announcement, put on a few, where am I? Yeah, I'm going to put a few things on, a few links in an announcement. So I just want to show you an example here quickly. Whenever you read literature, as you will for your projects, for example, you encounter all of these 3-letter abbreviations, and they're like some Latin or Greek term with super, infra, gyrus, sulcus, whatever, right? And you're like, okay, what does this even mean, right? What is this all about? And so I'm going to put up a few slides or links for you anyway that allow you to like browse through the basics of where these things are. So one example here is I just inflated this brain and you can start to have a look at where specific sulci, so these patterns of gyrification of the cerebral cortex, how they are organized in the human brain. Yeah. So I'll link to those so you can start looking at that if you want to look something up. Google helps also if you're uncertain about where things are in the brain, and you can always ask me. Yeah, I'll post that recording as soon as I'm able to record it. I hope they're just going to give me a new laptop or something. Thanks.
I didn't understand.